Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. We are from Group 2, consisting of 5 members. There are Regina Pramesti Santosa, Anti Apriliani, Salwa Maulani Sabila, Syalma Awalia Fitri, and I'm Saskia Fadilla Rachman as the moderator for today's presentations. This topic that will be presentation is cells. This topic is very important because cells is the basic units of life that makes up living things, including us. The main point that we will be talking about are firstly the cell theory and the cell history, secondly a typical animal and plant cell, thirdly chromosomes and mitosis, next the cell cycle and meiosis, finally we will be talking about microscope. Let's start with the cell theory and the cell history.
I appreciate the moderator allowing me to present the information. The history of cell is covered in the opening material. The smallest unit of a living item that can survive, like all living things, is the cell. Robert Hooke, an English scientist and inventor, made the discovery of the cell in 1665. The few features that were too small to be seen with the naked eye. Who created his own component like microscope. One of the first objects he looked at was a small piece of cork with its three other layers of bark. According to Hooke, the cork has the appearance of honeycomb and is composed of thousands of microscopic squares. These tiny boxes are what he named cells. It soon became clear that nearly all cells and that the cell serve certain characteristic command. The theory expressed by Hooke was then continued by Matthias Saintes and Theodor Swan in 1836. Then from this theory, it was proved that the cell is not an empty cavity but instead contains protoplasm, which is useful for supporting all activities of living things. The second material is about cell theory. The concept of the cell is the basic unit of life and is embodied in theory called the cell theory, which includes the following main idea. First, cell from the building blocks of living organisms. They are responsible for producing the energy that sustains life. The second is the cell effort only by dividing existing cells. The formation of new cell or daughter cell from pre-existing cell can occur through the process of cell division. Cell division is divided into three types: direct division, mitosis, indirect division, mitosis, and meiosis. The third in the cell contain inherited information and control their activities information that every living cell processes and that can be passed on its offspring. The fourth is the cell is the unit of life and function for metabolism, namely the semi-cell reaction of life that take place in the cell. The part of the cell that function of the site of metabolism in the cytoplasm. The cytoplasm is the full in cell that is located between the nucleus and the plasma membrane. The last that the cell is given the appropriate condition, namely that is able to live independently. The cell is called the smallest unit because it can be divided into smaller parts and each part can stand alone.
Thanks to Syalma for explaining about cell theory and cell history. Now, let's move on to the topic a typical animal cell and a typical plant cell.
Thanks to Saskia, let me elaborate on that. Typical animal and plant cell. First, a typical animal cell. Structure of a typical animal cell. First, the cell has a cell surface membrane. The cell membrane has a function to protect the cell. Second, the content consists of a central blood cell nucleus. The nucleus is surrounded by a fluid called cytoplasm. Third, the nucleus contains fibrous material. The fibrous material is called chromatin. The fourth, the condenses to form chromosome during cell division. 5. Chromatin contains DNA. DNA is the material that controls various activity the cell. 6. Scattered within cytoplasm are mitochondria. Mitochondria described as the powerhouse of a cell because they supply energy. The 7 smaller dots within the cytoplasm are particles of stored food. After we know to the structure of animal cell, now we proceed to the structure of plant cell. The first, most plant cells have a large cell-filled cavity called the vacuole. Salt is a watery fluid containing sugar and salt. The second, the cytoplasm contains starch grain. Starch granules are a product of plant food starch. The third, many plant cells have a chloroplast in the cytoplasm. Chloroplast contains the pigment used in photosynthesis. Chlorophyll, which is green, is the main pigment. Chloroplasts occur only in the part of the plant that exposed to light, the green part, usually found off leaf. Is the structure of an animal and plant cell. That's all from me. Return to Saskia.
Thanks to Salwa for explains about a typical animal cell and a typical plant cells. Now let's move on to the topic chromosomes and mitosis.
Thank you to the moderator for giving me the opportunity to continue my presentation. Introduce myself. My name is Regina Pramesti Santosa. The purpose of the presentation is to make a point about chromosomes and mitosis. Let's start with chromosomes. Are the filamentous bodies present in the eukaryotic nucleus? Chemically, eukaryotic chromosomes is made up of DNA, histone, and non-histone protein. Let me go back to what I said earlier about chromosomes. Firstly, chromosomes are the from DNA becomes in the nucleus when the cell is preparing to divide. Secondly, humans have 46 chromosomes, one set of 23 chromosomes from mom and one set of 23 chromosomes from dad. I'd like to illustrate this by showing you transparency chromosomes. Let me elaborate on that. One from nucleus. Chromosomes are loosely coiled strands of DNA and protein. After that, the second DNA doubles. The third DNA and protein coil up tightly, visible two copies of original chromosomes. Finally, there are copies 1 and copies 2. Now, let's move on to chromatid, seen in the picture on the side. Two exact copies of chromosomes that are connected together. The point where they are connected near the middle is called the centromere. Chromatids are made when new cells are going to be made. Regarding the next point is each species has a specific number of chromosomes, diploid 2 and 2 set of chromosomes found in all the non-sex cells or autosomes of an organism's body. Haploid and 1 set of chromosomes, only sperm and egg cell or sex cells have the haploid number. Let's start with the mitosis stage 1. Each duplicated chromosome appears as two identical or distinct twin chromatids. The chromosome condense and thicken. At this stage, the chromosomes are long, thin, and thread-like. The mitotic spindle begins to form. Its job is to grow the spindle between the centrosomes as they move apart. Two chromosomes gather at the equator on the metaphase plate. At metaphase, the spindle has captured all the chromosomes and lined them up in the center of the cell, ready to divide. At this stage, the two kinetochores of each chromosome should be attached to the microtubulus of the opposite spindle pole. Third, the spindle fibers begin to contract. This starts to pull the sister chromatids apart. At the end of anaphase, a complete set of daughter chromosomes is found on each pole. For at telophase, the cell has almost finished dividing and begins to rebuild its normal structure as cytokinesis progresses. The mitotic spindle is broken down into its building blocks. Two new nuclei, one from each set of chromosomes, the nuclear membrane and nucleolus reappear. Mitosis is the process nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. During cell division, mitosis specifically refers to the separation of duplicated genetic material carried in the nucleus. When mitosis takes place, there is no cell growth and all the cell's energy is focused on cell division. Cell divides to form two daughter cells with retaining same chromosomes, results in the formation of somatic cell equal number of chromosomes present in the mother and daughter cell.
Thanks to Regina for explains chromosomes and mitosis. Next, I will explain about the cell cycle and meiosis. The cell cycle. There are three main stages in the cell cycle. The first is interphase, the second is mitosis, and the third is cytokinesis. Let's start with interphase. Interphase. There are four main stages in interphase. The first is G0 phase, the second is G1 phase, the third is S phase, finally the G2 phase. G0 phase. G0 phase is the preparatory phase. If someone says G0 phase is the resting phase, it is not quite right, because at this phase, the cell is preparing to enter for G1 phase. Next, G1 phase. G1 phase lasts for 10 hours, and at this phase, the majority of the cell activity occurs. The cell grows and carries out its functions. Then, in the S phase, DNA replication occurs. Last, in the G2 phase is the preparation for the mitosis phase. Mitosis. In this phase, chromatids containing replicated DNA are separated from each other and are redistributed as chromosomes in the nuclei of the two new daughter cells. There are four main stages in the mitosis stage. namely is prophase, metaphase, anaphase, and telophase. Then, in cell division, also called cytokinesis, the cytoplasm divides to form two Dogger cells. Now, let's move on to the topic meiosis. In contrast to mitosis cell division that occurs body cells, in meiosis cell division that occurs gamete cells and nucleus divide twice. So it can be seen at the picture if in the mitosis cell division the stage is prophase, metaphase, anaphase, and telophase. In meiosis cell division, the stage is prophase 1, metaphase 1, anaphase 1, and telophase 1. After that, it continues with prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis cell division is the basis of sexual reproduction. In contrast to mitosis cell division that produces two diploid Dogger cells, in meiosis cell division that produces four haploid Dogger cells. I'd like to turn to something completely different. If earlier we discussed about cells, the next topic is tool that can see cells, namely is microscope.
As said earlier, a microscope is a tool for producing enlarged image of object or specimen, and therefore microscopes are often used in research, both in education and health. Well, this research is to enlarge image of objects that are micro-sized or that cannot be seen with the naked eye, such as cells found in plants, animal, and human. Wow, really cool, right? Now, let's move on to the discussion about who is the inventor of the microscope. And so, you need to know that the inventor of this microscope is a scientist from the country of windmills. Which country do you think? Yep, that's right, the Netherlands. In the years 1632 to 1723, a scientist named Anton van Leeuwenhoek invented a tool called a microscope. And a bit of information so Anton is called the father of biology. Wow, the scientist is very extraordinary, right? Since the 18th century, many types of microscopes have been invented, but until now very commonly used words are written below. Let me elaborate on that. Firstly, in a compound-like microscope, also known as an optical microscope, the lens will refract light to give a magnified image of the object, and then for the power of resolution, light microscope can magnify object up to about 1500 times without losing clarity. The second, namely transmission electron microscopy. This microscope is used to study the detail of the internal structure of cells. And as for how it works, the electron magnet will bend the electron beam to focus the image onto a fluorescent screen or photographic film. This microscope can produce clear images that are magnified up to 150,000 times. And finally, namely the scanning electron microscope, the way it works is by reflecting electrons off the surface of the specimen. which is tied with heavy metals and producing an image of the entire specimen, such as cell, tissues, or even organism. So many presentation about microscope for me. Thank you and see you.
Thanks to Auntie for explaining about microscope. In conclusion, cell was discovered in 1995 by English scientist and inventor Robert Hooke. Animal cells and plant cells have differences, as plant cells have chloroplasts, but animal cells don't have chloroplasts. In cell, there are chromosomes that carry genetic information. Humans have 23 pairs of chromosomes. Cells have two types of cell divisions. First, mitosis cell divisions that produce two diploid Dogger cells. Second, meiosis cell division that produces four haploid Dogger cells. The cell cycle have three main stages. The first is interphase, the second is mitosis phase, and last cytokinesis. The cell is very small, so we need a tool to see cell. The tool is microscope. There are three types of microscope, namely the electron microscope, the transmission electron microscope, and the scanning electron microscope. So that's all our presentation. Thank you for watching and thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.